Organoid research is already outpacing animal testing in ways you've never imagined, and the most mind-blowing breakthrough sits at number 1, a secret most labs keep hidden. Imagine a tiny liver the size of a grain of sand that can process medication exactly like a human organ. Mini-liver organoids are engineered from stem cells and arranged in three-dimensional scaffolds, allowing them to express the same enzymes that break down drugs in the real liver. Researchers feed these microorgans a dose of a new compound and watch, in real time, how it is metabolized, generating data that often mirrors human trials more closely than rabbit or mouse studies. Because the tissue is human-derived, species-specific toxicities that would be missed in animal models surface early, saving both time and lives. This approach also slashes the number of animals needed for preclinical safety screens. Stay tuned for number 9, where a brain-like organoid predicts neurological side effects before a pill ever reaches a human.
Surprisingly, a handful of cells can mimic the complexity of a human brain enough to flag dangerous side effects. Brain organoids are coaxed from pluripotent stem cells to self-assemble into layered structures that resemble cortical regions, complete with functional neurons that fire electrical signals. When a drug candidate is introduced, scientists monitor changes in network activity using calcium imaging, spotting disruptions that would cause seizures or cognitive deficits in people. These microbrains have already identified neurotoxic compounds that slipped past mouse testing, prompting early redesign of molecules. By capturing human-specific responses, they reduce reliance on primates and rodents for neurosafety assessments. Next up, number 8 reveals how heartbeat organoids keep a pulse on cardiovascular risk without a live animal.
In the realm of cardiology, beating organoids are rewriting the rulebook. Engineers shape stem-derived cardiac cells into tiny spheroids that contract rhythmically, producing a measurable pulse on microelectrode arrays. When a new drug is added, any alteration in beat frequency, strength, or rhythm is instantly recorded, exposing arrhythmogenic potential that often goes unnoticed in mouse hearts. Because these organoids possess human ion channel compositions, they predict QT interval prolongation, a leading cause of drug withdrawal, more accurately than traditional animal models. This technology has already cut dozens of animal experiments from safety pipelines and accelerated the path to clinical trials. Coming up at number 7, we'll explore how gut organoids reveal digestive toxicity without a single rat.
Hidden in a petri dish, a miniature intestine can reveal how a chemical irritates the gut lining. Gut lining organoids grow a hollow lumen lined with absorptive cells, mucus-producing goblet cells, and immune components, faithfully recreating the human intestinal barrier. Researchers expose these structures to food additives, pharmaceuticals, or environmental toxins, then assess barrier integrity, inflammation markers, and nutrient uptake. Findings often differ dramatically from rodent studies, where metabolism and microbiome variations can mask human-specific reactions. By pinpointing irritants early, companies can reformulate products before costly animal trials begin. This method also supports personalized medicine, as organoids can be derived from individual patients. Stay with us for number 6, where skin organoids illuminate irritation and allergy testing without a rabbit's ear.
Few people know that a tiny patch of lab-grown skin can replace the classic rabbit eye test for cosmetics. Skin organoids are layered with epidermal and dermal cells, forming a barrier that reacts to chemicals much like real human skin. When a formulation is applied, scientists measure swelling, cytokine release, and barrier disruption, providing a direct readout of irritation potential. Because the tissue is human, it captures allergic responses that differ from rabbit physiology, leading to safer consumer products. Moreover, the process is rapid, results appear within days, cutting both time and animal use dramatically. Up next, number 5 showcases how kidney organoids model nephrotoxicity with astonishing fidelity.
Remarkably, miniature kidneys can now predict drug-induced kidney damage before any animal is harmed. Kidney organoids form glomerula-like structures and tubular networks that filter waste and reabsorb nutrients, mirroring human renal function. When exposed to a candidate drug, researchers track markers of cellular stress, protein leakage, and structural changes using high-content imaging. These organoids have identified nephrotoxic effects that rodent studies missed, prompting early redesign of compounds and sparing countless lab animals from suffering. The technology also enables patient-specific testing, as organoids derived from a person's cells can forecast individual susceptibility to kidney injury. Coming up at number 4, we'll see how lung organoids breathe new life into respiratory safety testing.
Deep beneath the surface of a culture dish, lung organoids recreate the branching airways and alveolar sacs of a human lung. By exposing these structures to inhaled particles or aerosolized drugs, scientists can monitor inflammation, mucus production, and barrier permeability in real time. This approach captures human-specific responses to pollutants that are often misrepresented in mouse models, where airway architecture differs significantly. The data have already informed safer formulations for inhalers and highlighted toxicities of certain industrial chemicals, all without a single animal breath. Next, number 3 reveals how iCup organoids provide a humane alternative for vision-related toxicity testing.
One of the most delicate organs, the eye, now has a lab-grown counterpart that can predict visual toxicity without a rabbit's eye. iCup organoids develop retinal layers, photoreceptors, and supporting cells that respond to light and chemicals. When a compound is applied, changes in electrophysiological activity and cell viability are recorded, revealing potential damage to vision that animal tests sometimes overlook due to species-specific ocular differences. This method has accelerated safety assessments for ophthalmic drugs and cosmetics, cutting animal usage dramatically. Up next, number 2 uncovers how immune system organoids model vaccine responses with unprecedented precision.
Few people know that a tiny cluster of immune cells can simulate the human body's response to a vaccine. Immune system organoids contain B cells, T cells, and dendritic cells arranged in a microenvironment that mirrors lymphoid tissue. When a vaccine candidate is introduced, researchers track antibody production, cytokine release, and cell activation, gaining insight into efficacy and potential adverse reactions. Because the system uses human cells, it captures nuances of immune signaling that animal models often miss, especially for novel platforms like mRNA vaccines. This technology speeds up early-phase testing and reduces the need for primate studies. Finally, number 1 reveals the most groundbreaking advancement: whole-body organ-on-a-chip platforms that could replace entire animal studies.
At the summit of this revolution stands the whole-body organ-on-a-chip, a microfluidic marvel that links miniature liver, heart, kidney, brain, and gut organoids through tiny channels, mimicking human physiology in real time. When a drug flows through the system, each organ responds as it would in a living body, allowing scientists to observe metabolism, distribution, toxicity, and efficacy simultaneously. Early studies suggest these platforms can predict human outcomes with comparable accuracy to animal trials, while eliminating ethical concerns and interspecies variability. By integrating sensors that report electrical activity, hormone levels, and metabolic byproducts, researchers obtain a holistic view of a compound's profile in a single experiment. This could herald an era where entire animal testing pipelines are replaced by interconnected human tissues, reshaping biomedical research forever.
We've journeyed through 10 awe-inspiring ways that organoid science is quietly eclipsing traditional animal experiments, from tiny liver chips to brain-like mini-structures that feel almost human. Each step shows how compassion and precision can walk hand in hand, reshaping the very foundations of biomedical research. If you love discovering how these living models are changing the world, hit like and subscribe so you never miss the next mind-blowing list we drop each week. Tell me, which of these organoid breakthroughs impressed you the most? Drop your thoughts below, and let's keep the conversation rolling.